the process of now, the other day, take out your books or a pair.
इसके बाद क्या कराया था?
So now, when we are talking about the other day, we had discussed about the sexual and the asexual methods. One of you will take out your notebooks. Take out your notebooks and let us study the different methods of sexual, asexual reproduction. Now, we study what? Asexual methods of reproduction. That is what we discussed the other day. Sexual, asexual methods of reproduction. That is what we had studied the other day. What were some of the methods? I think we just introduced the topic. We have to learn the major differences. Did we write a table on the differences of sexual, asexual modes? We had written. Mitosis, meiosis, that was a concept that we had discussed the other day. That is important in this part of the chapter. The first part of the chapter, DNA copying is important. You should know what is DNA copying, what does it lead to. What did we say? What is DNA copying? Duplication of the chromosome. Duplication of the chromosome also leads to DNA copying. What else we had come across in that part of the chapter? The first part of the chapter was in the concept of DNA copying, the chromosome duplicates, also meaning the DNA duplicates. And if the DNA duplicates, there can be errors in the method, errors in the DNA duplication process. What does these errors lead to? Errors in the DNA copying lead to variations. What are variations we studied the other day? What are variations? Galti. What are variations? I had repeated the other day. What are variations? How will you write? What is it? Galti se mistake. What are variations? What did we explain? What did I explain you that day? Variations are the small changes that takes place in an offspring, in an organism. Small changes that develop in an offspring as compared to the parents. Why do variations happen? Errors during DNA copying. So are these variations good for an organism, good for a species? Yes, if you talk about these variations, mostly these variations are good for a type of a organism because it is making the organism better adapted to the changing environment. Remember all these concepts you'll have to write. Makes an organism better adaptable, better suited to the changing environment. Are we able to understand that variations, is it disadvantageous also? And if it is a disadvantageous variation, will it be very common in a population? If it is a disadvantageous variation, will a disadvantageous variation be very common in a population? No. What was the concept I explained you the other day? Disadvantageous variation. For example, a genetic error a person is born with, that will be 1 in 50 birth. 50 में एक जन का होगा, 100 में से एक जन का होगा. So that is what I'm mentioning, that variations, can this be disadvantageous? Yes, but disadvantageous variations are always less in a population. How do we understand that a variation is advantageous or disadvantageous? अगर पूरा set of organisms में है, then it is advantageous. अगर few individuals में है, then it is disadvantageous. The other day I gave you some examples also. Again today I'm mentioning a person born with a birth defect of the lungs, of the heart. It is not common. Every individual is not born with that. Maybe 1 out of 40, 1 out of 50 birth is a case, something like this. So that is understood that this is a disadvantageous variation which will not allow the individual to survive for a long period of time, and it is not getting passed on to the next generations also. That's fine. So this was the concept that we had studied the other day. When we answer some questions, try understanding, try remembering this. Now next was a method of sexual, asexual, and we must have written down some differences between the sexual and the asexual methods. What are some of the major differences? If it is sexual method of reproduction, 2 parents involved. If it's asexual, 1 parent involved. If it is a sexual method, then in case of the sexual method of reproduction, the offsprings formed are few. Asexual, offsprings formed many. Sexual method, the method of reproduction is quite complex. Asexual method, the method is simple. And now we are going to come across, in quite a number of organisms, the asexual method is just a mitotic cell division, where the organism just divides or the cell divides into 2. A simple method. Number 3, I said. Number 4. What is number 4? If it is a sexual method, variations are definite. If it is a sexual method of reproduction, variations are definite. If it is a asexual method, variations are minimum. Because we are talking about the DNA of only 1 parent, asexual. So in that case, the offsprings are more or less identical to the parents. If it is asexual. Sexual, are the offsprings identical to the parents? No. We are different from our parents. Though we share a lot of similarity, we are different. What else? What is one major point I am not talking about? What does the process of meiosis give rise to? What was that word I told you the other day? Reproductive cells. What are reproductive cells also known as? Reproductive cells, what are they known as? Gametes. What are reproductive cells known as? Gametes. So the other day we discussed, if it is meiosis, meiosis gives rise to gametes, reproductive cells. And is there anything known as gametes or specialized reproductive cells when we talk about asexual reproduction? Nothing. In the method of asexual reproduction, there is nothing known as gametes or nothing known as specialized reproductive cell, fertilization, zygote formation. ऐसे कुछ भी concepts नहीं है. Let us learn some of those methods now. What are some of the methods of asexual reproduction? We'll first of all talk about some methods of asexual reproduction, but for certain methods. Now for all of these methods, there is diagram drawing as per your syllabus. So some diagrams I will ask you to draw now, maybe just 1 or 2, and some will be as homework. I will show you the specific figures. All figures are there in your textbook. You'll have to draw. The next class, definitely all of you will have to show me. Definitely all of you will have to show me. If you don't bring your diagrams completed, I'll make you sit outside and complete your diagrams again. What are the methods we are going to study now? Number 1. The methods that we are going to study now would be fission. Fission again of 2 types. One, binary fission. and the other multiple fission. Fission is, this is a concept, diagram drawing, very common to come in your paper. Binary fission, and the other one is multiple fission. So we are going to draw diagrams on this, binary and multiple fission. Apart from these two methods of fission, there are so many other types of asexual reproduction also. Which are the other methods of asexual reproduction? Let me talk about the second. The second method of reproduction would be budding. Let us talk about some specific methods. Budding. Budding, again, there are types. For budding also, there are types. Then apart from budding, we talk about fragmentation. We talk about fragmentation. Then apart from fragmentation, there is spore formation. These are the methods you have to study for. Let us go through all these. The first is binary fission. Common examples of organisms that show binary fission, all single-celled organisms, unicellular organisms, they show binary fission. One of the most common to talk about is amoeba, euglena, and paramecium. These are the organisms that show binary fission. So we talk about the euglena, we talk about the paramecium. All these organisms show the method of binary fission. How does this process of division happen? A single cell, that is the amoeba, irregular cell it is, always we are aware. This irregular cell, a single cell organism, what are the stages when you draw the diagram? Very simple. 2 marks for the diagram, very easy to pick up marks. Now, this particular cell, the first step is the elongation of the nucleus. So what is the next diagram that you'll have to draw? This is the cell and the nucleus gets elongated. This is the second step to draw. The elongation of the nucleus, and after that all of you will start drawing this diagram. Sharp pencil, no drawing with pen. Take out your pencils and start drawing this diagram. So now we are talking about elongation of the nucleus. This is followed by constriction of the nucleus. How will we show the constriction in the nucleus? The constriction of the nucleus has to be shown this way. This is the constriction that has happened in the nuclear region. The nucleus will become this way. By the time the nucleus is showing constriction, the cytoplasm also will start showing its constriction. So, nuclear and cytoplasmic constriction. Nuclear and cytoplasmic constriction has happened. What is the meaning of constriction? Some furrow formation in the process of getting divided into 2. This constriction will deepen further, resulting in the formation of 2 daughter cells. We can draw one more stage in between, or try showing the division of the nucleus this way, where the nucleus is just connected this way, the cytoplasm almost divided, constricted, and then 2 daughter cells formed, and then 2 daughter cells formed with the cytoplasmic division, nuclear division. Very simple concept. Draw your diagrams neat with the stages. Easily you can pick up 2 marks in this question. One complete diagram drawing is always there in the paper. So what are these known as? New cells. These new cells are known as daughter cells. What are these new cells known as? Daughter cells. What will this be known as? This is the parent cell. This is the parent cell. So we are talking about the division of the amoeba, single cell organism, to form 2 daughter cells. What are the features? What are the stages? The same thing you also get in your paper where the diagrams will be all mixed up. and you'll have to sequence them 1, 2, 3, 4. So 1, always the parent cell. 2, nucleus elongated. 3, nuclear constriction. 4, nuclear and cytoplasmic constriction definite, and then separation. So now, when we are talking about the characteristics of binary fission, what is it? Number 1, how many daughter cells? 2. Are the daughter cells almost identical to the parent? Yes. Essentially, it is identical to the parent. Is the parental identity lost or retained? That's a feature always you write. So we will write the parental identity is lost. What is the meaning? That is always written as a point of difference. Parental identity is lost. What is the meaning? Parental identity lost. Parental identity is lost, meaning now that we got the daughter cells, where is the parent? Is the parent parallelly existing? No, because it is the parent's body which got divided to form the daughter cells. So this concept is known as parental identity lost. In some of the other methods, you will come across that the parent and the new organisms parallelly are existing. For us, for like us, the offspring and the parent parallelly existing. Yes? So now when we are talking about this, these are the stages of binary fission. Done? All of you are done with the figure? Done with the figure? Now we talk about the next type, that is multiple fission. For multiple fission, what are the stages? For multiple fission, what would the stages be? And then after that, you should be able to write at least 2 differences. Multiple fission. What would the characteristics be? In case of multiple fission, generally we talk about organisms like single-celled organisms but which are pathogenic, meaning capable of causing diseases. And these are the ones which develop in large numbers. One common example, the Plasmodium. Malarial parasite, Plasmodium, and many more other protista, they generally show this method. Protozoans, they show this method. So I talk about the single-celled organism. This is the parent Plasmodium. This parent Plasmodium will undergo certain stages where the nucleus will divide into a large number of fragments. Multiple fission, hence the name. The nucleus dividing into large number of fragments, and every small fragment wrapped up by a small amount of the cytoplasm like this, and this is the pattern of nuclear and cytoplasmic division. This is the pattern of cytoplasmic and nuclear division, not clean like this. The cytoplasm also divided into many chunks. The nucleus also divided into many parts. जितने nuclear part, उसके आसपास उतने cytoplasmic division है. Then after that what? This will keep on increasing. These are the cell-like structures. This will keep on increasing. And once it is so many in number that they cannot hold, the membrane will burst out. The membrane will burst out. So now the structure will be something like this, where a large number of these structures are there. The nuclear structures, so many. And each one of these nuclei, these are known as nuclei. Nuclei, plural of nucleus. They are known as nuclei. So each of these nuclei now will be formed so many, wrapped up by a small amount of the cytoplasm. When the number is so many that the membrane cannot hold, the membrane will burst out. And this new nuclei are in the process of releasing out. And once they are releasing out, their growth and development into a new organism very much will depend on the favorable environmental conditions. So I would say that these are the new organisms, these are the new daughter cells in the process of release and in the process of growth to form a new organism. These are the offsprings. What are these? These are the nuclei, a large number of nuclei. But this nuclei also contains cytoplasm, and provided they get adequate amount of moisture, temperature, nutrients, they will start growing into a new organism again. So what is the prominent difference? I'm saying if it is a concept of binary fishes, in case of binary fission, the organism that shows binary fission is always unicellular, but the non-disease causing types of organisms always. The non-disease causing types. So non-disease causing types को क्या बोलेंगे? Free living organisms. Unicellular, free living organisms show this method. What about that side? Unicellular, but disease causing ones show that method. Multiple fission. Because they have to establish in the body of the host. In large numbers, they have to establish in the body of the host. So now, I would say, what are these type of organisms known as? Pathogenic, disease-causing forms. Two daughter cells, numerous daughter cells. Numerous daughter cells are there. Then, when we are talking about the nucleus and the cytoplasm, how many times the nucleus divides? One. How many times the nucleus divides? Once. Numerous, number of times, multiple times the nucleus is dividing, and surrounding every nucleus there is a bit of a cytoplasm, and that is the new organism, how it starts developing. So multiple fission and binary fission, the types of organisms, examples, and some one or two differences you should be able to remember. Let us write 2 points of differences very fast. Make a table. This is very common. Should not make any mistake here. Binary fission, make a table. Let us write the differences. Number 1, for binary fission, right? Nucleus divides only once. Binary fission, nucleus divides only once. Other side, multiple fission, nucleus divides multiple times. Nucleus divides multiple times, many times. Nucleus divides multiple times. Next, number 2. For binary fission, takes place in free-living single-celled organisms. Takes place in free-living single-celled organisms. Takes place in free-living single-celled organisms. Other side, for multiple fission, takes place in pathogenic. Takes place in pathogenic single-celled organisms. Takes place in pathogenic single-celled organisms. Takes place in pathogenic single-celled organisms. Number 3, 2 daughter cells formed. 2 daughter cells formed. And this side, numerous daughter cells formed. Numerous daughter cells formed. Let us go to the next method, budding. What is the concept of budding here? Again, we are talking about a single-celled organism or a multicellular. We are talking about a single-celled organism or a multicellular. Now, budding can happen in different organisms in different way. I may talk about a single-celled organism. Yes, if it is a single-celled organism, then I think of yeast. What is yeast? What type of organism yeast? Yeast, what type of organism? Fungus. Yeast is a fungus. So now, if I have to think of budding single-celled organism may, then yeast. If I have to think of budding in multicellular organism, I think of budding in multicellular organism, then Hydra. And I talk about budding in plants. Possible hai budding in plants, yes. How budding in plants? Bryophylla. So now we are talking about the same concept of budding, but then in different organisms in different manner. Let us learn one by one. So if it is a concept of budding, what is happening here? If it is a concept of budding, the first one that I am talking about, a single cell organism, yeast. So yeast में budding कैसे होगा? How is the process of budding happening in the yeast? In the process, a single cell organism, this is the parent organism. Very simple organization, nucleus है, proteins है, cytoplasm है, and the parent cell will give rise to a small bud. What is the bud? A region where certain amount of cells have gathered and rapid cell division. A bud. This bud will grow. This bud grows. By that time, the nucleus has divided. The cytoplasm has divided, but equal है cytoplasmic division. Cytoplasmic division इसके जैसे equal है? No, unequal cytoplasmic division. Nuclear division is almost equal, but cytoplasmic division is uneven. The nucleus is wrapping around a small part of the cytoplasm membrane बन गया, and that is the new organism in the process of formation. But now that the new organism has formed, in most of the cases, this new organism will not detach from the parent body, that will remain attached to the parent body. And dhire dhire, a chain of buds will be formed. Something like this. A chain of buds. Okay, I don't have a figure for that. I'll show you here. I talk about the concept of budding. If I am talking about a concept of budding, and for the yeast I'm thinking, and I'm saying not necessarily every time the new organism will get detached. I understand this is a single yeast and this is the nucleus in the process of budding. How will it happen? Equal nuclear division, unequal cytoplasmic divisions. And how it is happening there? This organism is forming a small bud, nucleus divide, cytoplasmic unequal division, and this is progressively growing, and in few seconds it has developed into a complete organism. Ho gaya. Then it is supposed to detach from the parent body, no, but it does not. It will not detach from the parent body. Ab jab growth complete ho gaya, it is not detaching from the parent body, but it will again result in the formation of a small bud. And then this nucleus again dividing, and the nucleus will come here. So after some time, what now? Again a new structure develops. So this type of yeast you get to see when we talk about in the entire process of making of the cakes, bun, pastries, etc. These are actually, we get to buy them from the market, etc. in the form of fine thread-like structures. Put it under the microscope. These are long chains. Long chain of buds like this. So this is the new organism. This is the new organism here. This is the new generation yeast. This is the new generation yeast, new organism, and this is the parent organism. एक से दूसरे, दूसरे से तीसरे, and then they will form a long chain of buds, and sometimes they will detach. But not possible every time. One bud forms, grows, detach. No, not that method at all. That is yeast, single-celled organism. Talk about now next, multicellular organism में कैसे? In case of a multicellular organism, the method is like this. This is the Hydra. Did you ever come across this word Hydra? Where is Hydra found? Huh? Water. The lake, pond, deep sea water bodies में नहीं. generally the shallow water bodies. So this hydra, can we see the hydra with our naked eyes without a microscope? हाँ देख सकते हैं ना? Detail में नहीं देख सकते हो, but then you can see the hydra in the form of a fine thread. And that fine thread you put under the microscope, तब जाके we can see the detailed structure this way. So this particular hydra, how did the budding start? Why the word budding? Budding from the word, this mature organism, it is a multicellular organism. It is a multicellular organism. It will give rise to a small bud. What is a bud? An accumulation of fast dividing cells. So from this bud, process of cell division starts. Formation of new cells and tissues starts, utilizing the nutrients of the parent body. And then, in a short period of time, this bud starts growing into a complete organism with those few tissues and structures. It is a multicellular organism, but it is a very simple organism. Does this organism have any organs in its body? Organs, when I say, nothing. They have got just few tissues. Just a handful of tissues for respiration, excretion, that's it. So simple they are. So definitely by the way of budding, a new organism can easily be formed. Simple cell division ही चाहिए and a new organism would develop. Necessary है एक ही बने? No, it can be multiple also. 5, 7, 8. Potato bud is not a asexual method of reproduction. वो अलग है, vegetative propagation. We will learn that later. So हाँ, नहीं. When you are talking about the concept of potato, that is vegetative propagation. Spore formation is the asexual mode. Vegetative propagation अलग है. Vegetative propagation मतलब certain plants they don't give rise to flowers, and from the plant part they start growing a new plant. That is vegetative propagation. So now I am talking about budding right now. It is a method of asexual reproduction. So first of all, we talk about the animal types. I gave you one example of the yeast. Yeast also show budding. A chain of buds can be formed. We talk about multicellular organism like hydra also can show budding, buds, grow, develop, cell division, form the few tissues, and then the entire organism. But this entire organism, as long as the full body growth is not happening, the entire organism will not detach from the parent body. Even if the entire organism detaches from the parent body, the new generation organism and the parent are existing together. So we will say, for budding, the parental identity is lost. Is it lost? Parental identity, is it lost? No, it is not the parent body dividing to form the organism, a part of the parental body dividing to form the organism. So for budding, it has to be parental identity is retained. Parental identity is retained. Meaning क्या है? Parental identity retained मतलब if the new organism and the parent can exist together. Fission में we had seen parent and new organism cannot exist together. So इसीलिए we had written lost. Parent and new organism they never exist together. तो ये हो गया in hydra. For a plant also we now see, but that is actually a concept of vegetative propagation, but I will take it together. To understand the concept of budding in a plant, how budding in a plant, I have it here. How budding in a plant? This is a bryophyllum plant. This is a bryophyllum. This type of plants, these are short plants. The leaves will always produce such type of buds at the margins. The leaves will make such type of buds on the margins of the leaf. And these buds, if they detach from the leaf and fall on the soil, or quite a number of times, this has given rise to some adventitious roots. Can you see? The buds will give rise to some fine root-like structures. How tall are these plants? Short. From the level of the soil, they will be hardly this much. They are not tall trees at all. Tall plants, we are not talking about this. These type of plants are majorly found in all gardens. And these are short plants, branching. Something like the leaves are almost like those money plants, thick, fleshy. And they will always develop buds like this. And in just 1 or 2 days, you get to see that the buds start developing these roots. The moment these roots start touching the soil, when the leaf has fallen from the plant, the roots will come in contact with the soil, and from this, a new plant will develop. Quite a number of times, the buds would detach from the leaf, come in contact with the soil, new plant develop. Quite a number of times, the leaf has so many buds, and now it is detaching from the plant. The roots come in contact with the soil, new plant develops. So these are the 3 different methods of budding that we had come across. We talk about fragmentation now. What is fragmentation? Why the word fragmentation? In what type of organisms fragmentation? Can fragmentation happen in multicellular organisms? Yes, only in multicellular organisms. Do we ever come across a concept of fragmentation in unicellular? No. So fragmentation, to talk about some examples, again, we are talking about examples like the Spirogyra, the Obelia, the Spirogyra, again present in the lake and the pond and the shallow waters. So what would happen if it is a concept of reproduction, but we now try to understand asexual reproduction. This type of an organism, this is known as the Spirogyra. How is their body shape? Filamentous, ribbon-like structures they have. This type of Spirogyra on the water, I talk about the water body. Every type of a water body has got its characteristic color, green, blue. This is majorly because of this type of organisms present in the water in large numbers and the algae present in. So now, this type of organisms, where are they present? They are present in the superficial layers of the water body. So collect some amount of the water, put few drops of water in the slide and observe under the microscope, we will get to see them. Naked eyes, are we able to see them? No, we are not able to see them with the naked eyes. But in the water, we can see that the water is little bit dirty, the water is little bit turbid. Put that few drops of water on the slide, observe under the microscope, this type of structures, large numbers. Spirogyra, how is their structure? Long, ribbon-like, generally greenish because they have got chloroplast in them. This organism, when it is fully formed, what will happen? This entire organism, the Spirogyra that I have to remember, this organism which is long, slender, filamentous like this, and I just now say that they are multicellular. A single mature organism may have 20-25 cells. Organs present? Nothing, just few cells in their body. So this organism will have a number of cells like this, will have a number of cells like this, multicellular of course, but no definite tissues and organs. And they have got the chloroplast I mentioned. Sits right. They have the chloroplast I mentioned. So they will be having these threads of chloroplast all over. They will be having the threads of chloroplast all over, making the entire structure look green. In the water they are present. But once this entire structure organism is fully mature, what is this ribbon-like structure known as? This ribbon-like structure is known as the thallus. Thallus क्या है? The entire filamentous structure of the organism जो absolutely mature हो चुका है. अब जब ये पूरा mature हो चुका है, normally the entire organism will divide into fragments. Normally. It is not an accident that we are talking about. It is no predation we are talking about, nothing. The entire organism, once fully developed, breaks down into few fragments. Hence the name fragmentation. अब हर एक fragment में if there are adequate number of cells, so for example I would say that ये जो fragment है, this will definitely grow into a new organism क्योंकि इसमें full cells almost है. So if there are adequate number of cells, yes, this will grow into a complete full organism. Will this grow? Okay, this will also grow into a complete full organism. What about this? If I cut it little bit, no, it does not have full adequate number of cells. So this may not give rise into a complete organism. So that is the concept. This entire spirogyra, into how many organisms will it give rise to? Kuch confirm rehta hai? No, nothing is confirmed. When the body divides into few fragments, does the fragment have adequate number of cells? If yes, it will give rise to a new organism. If no, it will not give rise to a new organism. And that's how sometimes we may come across 5 organisms from one filament, sometimes 8 organisms from one filament. So it varies from time to time. This is fragmentation. What type of organisms? These are also semi-aquatic organisms, shallow water organisms. Which example we are talking about? Spirogyra. Okay. Now, this is the concept of fragmentation. Now, if I have to write down one difference between budding and fragmentation, what are the two differences to mention? Budding and fragmentation में अगर दो difference लिखना है, what should you write? In budding, accumulation, the process of growth starts by the process of accumulation of the fast dividing cells. How does the division start? Dividing of the thallus into segments. वो नहीं चाहिए. Those words no need for you. So now when we are talking about this, how does the method start? Is the number of organisms formed definite? And is the parental identity maintained or lost? Based on this only, you should always write the differences. Clear? Let me give you one example. अब इसमें तो काफी सारे methods है. I will not give you every difference. Concept जो समझ में आया है, based on that you will have to frame your answer. Let me just give you a format कैसे लिखते हैं. Let us take difference between budding and fragmentation. So on what points you should think of writing down the difference? Budding and fragmentation. What are the differences? You will keep space for drawing diagrams, or else you complete now and later you draw diagrams. I'll give you a proper question. Draw diagrams neat with pencil and show me the next class. Right? Huh? Achha, drawing now. Okay, fine. Those of you who haven't drawn, you'll have to show me all these diagrams drawn. Abhi answer likh lo pehle.
Fragmentation and regeneration is same.
No. Fragmentation and regeneration is different. I'll tell you. Okay, first लिखते हैं fragmentation and budding. Fast. Fragmentation takes place in simple multicellular organisms. Takes place in simple multicellular organisms. Budding takes place in multicellular organisms. Budding takes place in single cell and simple multicellular organisms. Single celled and simple multicellular organisms. Number 2, organisms. Number 2, for fragmentation, thallus divides. You have to remember what is thallus, the full grown filament. Thallus divides. This is the entire thallus I showed you earlier. Thallus divides randomly into few fragments. Thallus divides randomly into few fragments. देखो Thallus divides randomly into few fragments. Other side, for budding, specialized cells, specialized fast dividing cells, specialized fast dividing cells accumulate at different regions of the parent body. At different regions of the parent body. It can be 1, it can be 5, it can be 7 based on the organism. Different regions of the parent body. Different regions of the parent body. Last point. Buds complete for budding. Buds complete their growth remaining attached to the parent body. Buds complete their growth remaining attached to the parent body. Remaining attached to the parent body. Budding. Buds complete growth remaining attached to the parent body. Each fragment completes its growth remaining attached to the parent body. Remaining attached to the parent body. Fragment. Each fragment completes its growth to form the entire thallus. Each fragment completes its growth to form the entire thallus. All points are clear? Did you all write it correctly? Likha? Last point. Budding. For budding, parental identity is maintained. For budding, parental identity maintained. For fragmentation, parental identity lost. Parental identity is maintained. So parental identity maintained for budding. Parental identity lost for fragmentation. All of you got the points? Now the next, we talk about the last method, that is spore formation. We are talking about modes of asexual reproduction. This is the last method that we are talking about. Spore formation. What type of organisms show spore formation? A large number of fungi. What are some common examples to write? Rhizopus. What are some common examples to write? The mold and the mucor and the mushrooms, all of these. Now that the monsoon is still on, I think all of you must have come across this Rhizopus. देखा? कौन सा वाला? Rhizopus देखा किसी ने? कहाँ पे देखा? हाँ? So now that we are talking about this, the fungus, the spore formation. If you open, ये वाला चाहिए, and वो थोड़ा सा और ना broad होगा diagram. Okay, next class I'll tell you that once all of you draw. Now this spore formation is a method. The spore formation is a method where we come across the fungus generally show this method. Fungal growth very much favorable when temperature is low and there is enough humidity in the air. So if there is enough amount of moisture and temperature is low, that is a suitable environment for fungal growth. And that's the reason whenever we are talking about the monsoon time, we often get to see fungal growth all over. We get to see fungal growth on the moist walls. We get to see fungal growth on the moist leather items. If you open your shoe cupboard, there must be some few pairs of leather shoes. If you take them out, you'll get to see that it's all covered with some whitish, granular structure all over, and with a foul smell. Did you ever notice? This is all rhizopus. Where do we get to see the mold on the bread? Any type of bread, pastry, cake, any type of these items which are baked, which has got enough amount of moisture in it, keep it outside. Don't refrigerate. Just 1 day is enough. At the most, if the weather is like this, 2 days enough. बहुत ज़्यादा बारिश हो रहा है, 1 day is enough, and you will get to see on the surface of the bread some blackish or greenish cottony growth has happened. Some cottony growth will happen all throughout on the surface. Cottony growth has happened. That is all the mold, also known as the bread mold. So now, when we are talking about this, what is it? आज कौन सा विषय है? क्ले पे, हाँ, क्ले के, हाँ, वो अभी फेंको उसको. Clay, the fungal spores will never get cleared out. It will remain there. It will remain there. Clay is something, this molding clay में fungus बहुत जल्दी आता है. बहुत जल्दी, मतलब because it is already moist. That's the reason you are able to mold it. So you have used it once, twice, abhi straight away you'll have to throw it away. Because it will have enough amount of fungus and that causes rashes. If your skin is sensitive, it causes major rashes on your skin. So when we are talking about this, the fungus, now that I'm talking about the bread mold. Now how does this process of reproduction happen? I talk about the rhizopus. Look at this figure. The rhizopus, the structure is something like this. This is the mushroom that we are talking about. Let us talk about the other one first. Mushroom. That's a mushroom. Now, this is the structure of the rhizopus. Just now I was mentioning, that in your leather items, the leather bags, leather shoes, you see, which you are not wearing for the last 1-2 months. Take out and observe. You will see some white cottony growth there. This is the rhizopus. But what has happened? If you observe them clearly under the microscope, you will get to see that on the surface there are some fine filamentous structures. These fine filamentous structures, these are all multicellular. Inme se one of these cells, they have started showing almost a similar method like multiple fission. And then resulted in the formation of spores. These spores will all accumulate, and once the conditions are favorable, what will happen? These spores, again same like multiple fission, the membrane is now absolutely filled with the spores, membrane will break up. The spores are all released. Now here you'll have to remember a few words. This entire knob-like structure which is containing the spores, what is it known as? And the spores, what is it generally meant for? So if I talk about the proper, this is the diagrammatic representation of spores, but whenever we are drawing the diagram, this is the entire filament. This is the entire filament actually, and this is all multicellular. What is this entire multicellular structure known as? This multicellular structure which has still now not started producing spores. What are we going to name this as? This will be named as the hypha. All of you will draw this diagram because this is a bit different from this one. This diagram you'll have to remember. This is a multicellular structure. Now this, one of these cells, they will start dividing, showing a method quite similar like multiple fission. So what is the next diagram to draw? This one, when we are talking about the next diagram here, would be that one of these cells, they have started showing a method of multiple nuclear division. They have started showing a method of multiple nuclear division. Something like this you also get to see on the bread mold. Small black, brownish, greenish, small dots. And then some fine cottony threads. This is the cottony thread, this is the mold. So what has happened? This type of large number of spores have developed, and this is the structure there. Large number of spores have developed. This is the structure. You'll have to remember the names here. What is this name now? I talk about this knob-like structure which is containing numerous spores. What is it called? This is known as sporangium. Take a note of this word. The knob-like structure containing millions of spores. What are these called? And this granular structure, these are known as spores. This entire knob-like structure, what is it called? Sporangium. What is it containing? Spores. So when the spores are too many now, what will happen? The spores will be all released out like this. The spores will be all released out like this. These spores, the fungal spores are almost every time there. The fungal spores are always there in the air. They are so light. They are so small. We cannot see them with our own eyes. We cannot see them with our naked eyes. We need a microscope to see that. So these fungal spores, whenever they deposit on a suitable area, moisture, temperature, etc., nutrients, then they start growing again, something like that. So this method of spore formation is very rapid, very rapid method. That's why fungal infection, the dandruff that we suffer from, that is also a fungal infection, spore formation. Yes. So, abhi question 8 likho, all of you. You are supposed to draw diagrams for fission is already over. Draw diagrams for neat, clear pencil diagram. याद रखो. Diagrams for fragmentation number 1, budding number 2, spore formation number 3. अगले दिन मुझे दिखा. Budding, fragmentation, spore formation. ये 3 मेथड का ड्रॉ करना है. Budding, fragmentation, spore formation, neat, clear diagrams draw करना है. बनाया था क्या? कंप्लीट करो एंड नेक्स्ट क्लास यू शो. Heredity पढ़ो. If there is any doubt, clarify. How many of you had read that chapter properly? Which अभी तक भी start करना है. Heredity. How many of you are okay with that chapter, girls? Chapter syllabus में है, पता है ना? Syllabus में है, पता है क्या? Did you read that chapter, Heredity? Are the laws process clear to you? पढ़ो.